So, Melanie, I want to turn the discussion to you because I know you have a background in mRNA's cousin industry, in the oligonucleotide space, right? So it's a little bit different than what we're working with in terms of a manufacturing paradigm, but it is a bit more mature from a regulatory perspective and a commercial perspective. So I'm curious to get a sense of what learnings, if any, right, we can take from that side of the industry, even though it is CBER. Are there collaborations between the two offices that you think are going to be helpful? What can we learn from what our cousins are doing over there? And should we even be making the comparison, right?
Yeah, well, I mean, certainly when it comes to gene editing, where there's a guide involved, you're going to have the two cousins mixed together in an LNP, and so it does help us to think about both nucleic acids. I would say the one thing that was I come out of the oligo world, and for me, my favorite tool is a mass spec. I can't use that for mRNA in a way that's similar, right? So for oligos, understanding your impurity profile, your product-related impurities is much easier. You can understand what they are. You can determine structure. You can look at batch-to-batch consistency of specifically defined molecular weight species. We can't really do that as well in mRNA. And the tool that is the mainstream tool right now for looking at purity of mRNA is this capillary electrophoresis, which is this really coarse tool because you can't fractionate it. You can't, we can't put the mRNA in a mass spec. So really understanding the product related impurities that are associated with each batch that you made is currently elusive and needs a lot of work. So I guess what I would say, and it could be that these product related impurities don't matter as much as they do on the CBER oligonucleotide. That could well be the case from a safety perspective, but from a process characterization perspective, to know that you're doing the same thing from batch to batch, understanding those species is important for consistency. So I would say that's one of the things that I'm quite keen on, working on and seeing how that advances for sure. And I will say that anecdotally, I have heard from the gene editing space, CBER is more interested in the oligo purity of the guide, more so in a manner that is consistent with the way they're interested in it in CBER. Than the mRNA. I shouldn't say more concerned, but we see more effort being put into the characterization on the guide than the mRNA.